What happens when a point mutation changes the start codon during protein synthesis? In this lesson, we'll explore what happens when the critical start codon August gets mutated to a different codon. First, let's understand what makes the start codon so important. AUG is the universal start codon that signals where translation should begin on the mRNA strand. This codon not only signals the start, but also codes for the amino acid methionine. Here's an mRNA strand with the August start codon highlighted. The ribosome recognizes this sequence to begin translation and adds methionine as the first amino acid. Ribosomes specifically recognize August as the signal to initiate translation, making it essential for protein synthesis. Let's review how normal protein synthesis works with the correct start codon. Here's an example mRNA sequence starting with AUG and ending with a stop codon UAA. With the correct start codon, translation begins at methionine and continues to build the polypeptide chain with arginine, phenylalanine, and glutamic acid in sequence. Now let's see what happens when a point mutation changes the start codon. A point mutation is a change in just one nucleotide in the DNA sequence. The start codon August could mutate to AUA, AUC, or AUU by changing just the last nucleotide. Here we see a point mutation changing the G in August to an A, creating AUA instead. This single nucleotide change has major consequences. The consequences of mutating the start codon are severe and immediate. First, the ribosome loses the signal telling it where to start translation. The ribosome cannot recognize AUA as a start signal, so it fails to initiate translation at this position. The ribosome will continue scanning downstream for the next August codon it can find. The ribosome scans past the mutated codon until it finds another August downstream in the sequence. There are three possible outcomes depending on the mRNA sequence downstream. If there's another August downstream, translation starts there, producing a shorter protein missing the N-terminal region. The normal protein has 4 amino acids, but the mutated version starts later and only has 3, missing the original N-terminal methionine. If there's no other August codon downstream, no protein is made at all from this mRNA. When no August exists downstream, the ribosome never initiates translation, resulting in complete loss of protein production. If a downstream August exists, but in a different reading frame, a completely different, likely non-functional protein is made. Let's understand the broader impact on the cell. Without the correct protein, cellular processes that depend on it will be disrupted or fail completely. The cell's normal functions become impaired as critical proteins are missing or non-functional. The severity of the impact depends on how important the affected protein is for cell survival and function. If the protein is essential for survival, the mutation could lead to cell death. If the protein is non-essential, the cell may survive, but with reduced function. These mutations have real consequences in genetic diseases. While start codon mutations are relatively rare, they are devastating when they do occur. If the mutation affects a critical gene, it can cause serious genetic diseases. Possible outcomes include enzyme deficiencies, metabolic disorders, and developmental defects depending on which protein is affected. Let's summarize what happens when the start codon is mutated. The AUG start codon is absolutely essential for the ribosome to know where to begin translation. A point mutation changing the start codon prevents normal protein synthesis from occurring. The result can be a shorter protein, no protein at all, or a completely wrong protein. The cellular consequences vary based on how important the affected protein is for cell function. Remember, the start codon is critical for life. Protecting it from mutations is essential for proper protein synthesis and cellular function.